The question is, describe Mendel's procedure of conducting a monohybrid cross in pea plant. Monohybrid cross means the parents differ in one genetic trait. So the process involves the following step. First is the selection of two sets of pure breeding pea plants with contrasting characters, for example height or flower color, flower position show, so on. Now if we take height as the character, the contrasting characters will be tall and dwarf. Pure bred lines means they both are homozygous carrying same type of allele. So tall plants were 183 centimeters or 6 feet in height, while dwarf or short plants were 30 centimeter or 1 foot tall. He called the tall plants as homozygous tall, having capital T alleles, whereas dwarf plants as homozygous dwarf, having small letter t or lowercase t. These plants were called as parental generation or represented by P. Now, the first step is to do the cross pollination. So he took the tall plants and crossed with dwarf plant, and this involves two different parents, hence known as cross pollination between two different plants. Now the seeds produced by cross pollinating these P generation plants were collected and grown to produce hybrid plants. Now if we see the first generation or the first filial generation, they all were tall. Now if we see the genotype, they have homozygous tall and crossed with homozygous dwarf, and tall character is dominant over the dwarf trait. So all the F1 generation plants were tall and all of them were heterozygous, having one capital or the dominant allele and one the recessive allele, that is for dwarf. The plants of F1 generation were allowed to self-pollinate and seeds were collected. Now the plants raised from these seeds obtained from F1 plants belong to second filial generation, that is F2 generation. Now Mendel made a note of how much of them or how many numbers were tall and how many number of plants were dwarf. Now Mendel noted it, 75% of plants of F2 were tall, whereas 25% were dwarf. He concluded that F2 generation, the tall and dwarf plants were produced in 3:1 ratio. So tall, that will be this heterozygous tall, one homozygous tall and one heterozygous tall. So 3 tall and 1 dwarf. This is phenotypic, that is what is visible to us. Now Mendel raised F3 generation from the F2 plants by self-pollination again and found that dwarf plants of F2 generation produced only dwarf plants on self-pollination, because if we see they are homozygous, and if I self-pollinate, the next generation will get the same trait. Whereas if we see F2 tall plants, there are two possibilities. One is homozygous tall, other is heterozygous tall. Here only 25% were pure tall and produced tall plants, whereas the remaining 50% plants produced both tall and dwarf plants, again in the ratio 3:1. That means it is equal to the cross that we saw, that is F1 generation cross. So genotypic ratio was later concluded to be 1:2:1. I hope it is clear to you. Thank you.